Okay. Good afternoon. Thanks, Melissa, very much for this fantastic introduction. And I also want to express my special thanks to Jean and the organization committee for giving me this opportunity to present our research, which daily happens in the BRB building next door, and to the broad audience. Okay. Oh, I will just do that. Is that better? Okay. Good. So I'm very fortunate because I think our research really fits in the topic of the symposium, Understanding Life. So what we are interested in BRB, a group of researchers, we do want to understand how and why we age. So I guess that's. So we consider ourselves as aging researchers, or human aging researchers. This can be a little bit challenging because of the time-consuming aspect of the nature of the questions. So how we scientists are going to address these type of questions? Commonly, model systems are used. A bunch of model systems are showing here. So as this one. Okay, this is a pointer. So this is a single cell yeast, so has been used and reviewed a lot of molecular insights of normal human aging. And the C. elegans as invertebrate models. Then the mammals, obviously, the mouse is a very popular model. And here I just want to tell you, maybe in the near future, I will be able to present you a collaborative work from my laboratory and Dr. Zhongqi Liu from my department. So we started to understand the aging process in plant. We use a flowering plant as a system to study it. But today, what I will be telling you is what our study using the rare human conditions called Hutchinson-Gilford Progeria syndrome. What we have learned in the past few years from this syndrome and what are the molecular connections from Hutchinson-Gilford Progeria syndrome, which in short is HGPS, to the normal human aging processes. And my talk is going to focus on the recent studies. And the recent, I mean, we published pretty much in the last 2 years and some unpublished data. So what's progeria? As Melissa already introduced you, it's a premature aging or accelerated aging syndrome. So if you are looking at a picture up there, so it is I like to show it again and again because it's a progeria patient holding his own pictures at different ages. So what you can see when the patient was very young, 1 or 2 years old, he looked pretty normal. But when he grew up, you can see the rapid aging syndrome started to show the clinical features, including the loss of hair, the diminished subcutaneous fat, and there is a clear growth retardation. The skeletal abnormalities are very clear, as you can see from the hands. And most importantly, the progeria patients, they die early. They die in their early teens due to cardiovascular disease. So what do we know about this rare human disease? About 9 years ago, so Maria Erickson and Francis Collins, they set up a screen. They pull up many progeria samples and sequenced, try to identify what is the gene causing progeria. To their surprise, more than 90% of the samples they collected showing one single nucleotide mutation in the coded region of a non-protein. This protein is called lamin A. So they were happy, but then I always want to ask them, are you guys disappointed when you find out that this coding region actually falls in the third of the amino acid coding? So if we learn biology, we all know, usually they do not change the coding sequence. So they name the mutation glycine to glycine mutation, G608G, because 608 is the position of the nucleotide, the amino acid sequencing. They were probably disappointed for a couple of days. So what happened next? They aligned this sequence carefully and looked at it to the common splice donor sequences. So what they have found is the common splicing donor sequences has 7 consensus nucleotide. And here, they have 6 out of 7 consensus to the common splice donor site. So they hypothesized that this mutation can activate a cryptic splice site in the lamin A gene and lead to an internal deleted mutant, which is a mutant lacks the 50 amino acids here. Is that true? So quickly, actually, people using the Progeria skin fibroblast samples, they were able to detect a band which migrates a little bit smaller than the wild-type lamin A, which we call it now progerin. The progerin has an internal deletion, lacks the 50 amino acids, but still have the similar C-terminal, exon 12, the C-terminal ends. So now we find the mutation. The next problem is what's the function of lamin A? Why it causes such a drastic syndrome? So when we look at here is a diagram. I first want to orient you to the diagram. So here is a nuclear plasma, and on the top is a cytoplasm, and this is a nuclear basket. Where is the lamin A located? So the lamin A, together with its brothers and sisters, lamin B and C, are actually located in right underneath the nuclear envelope in a greenish meshwork structure showing you here. So the lamin A has been proposed to form a meshwork structure and give the structural support to the nuclear envelope. Indeed, what happens to the Progeria fibroblast cells? When they look at it carefully, they find out in the normal scenario, we see the nucleus look like a perfect oval shape. But what happened to the Progeria cells? You see the Progeria nucleus become very abnormal, so some imagination, some blabbings. We give it a cute name called nuclear blabbing. So, but in addition to that, what happens to the lamin A? Do they have other functions? So we see the lamin A actually interacts with a variety of factors, as showing you here the chromosomes, and also the chromosome is in the yellow line, and also a transcriptional factor called RB and other important transcriptional factors. So then the current working hypothesis in the lab, we propose progerin causes the cellular damage, not only by its structural effects, but also by its way it alters the cell, the chromatin organizations and the transcription regulations. How are we going to test it? I first will tell you a little bit about our studies on the nuclear structure. Since the nuclear structure as a primary cellular phenotype, and now as a scientist, we kind of know why the Progeria nuclear structure is abnormal. In order to understand this, we have to go back to look at what happened to the lamin A when it is processed in the cell. So when the lamin A is first made, it is called pre-lamin A. It has a little tail called CAAX in the end, and the cysteine is going to be farnesylated, which is a process, lipid modification process. After farnesylation, there are a couple of other additional steps. Then a proteinase comes and cleaved. There is a conserved ISY-LLG domain. So once after its cleavage, we get a mature lamin A, which is shorter than the pre-lamin A, but without the purple farnesylation. What happened to Progeria cells? So because I already told you there is an internal deletion, However, the C-terminal is retained, so there is still a CAX motif. The farnesylation happened. Then the other steps happened. But the cleavage site is missing. It's in the deleted 50 amino acids. So the progerin still maintains the C-terminal lipid modification. It has been hypothesized this modification can insert the progerin abnormally into the nuclear membrane. That's why it is causing the nuclear blabbing phenotype. Is it true? So scientists designed another experiment here to use a drug called farnesyltransferase inhibitor. So this is actually an anti-cancer drug. So they use this drug to block the first step of the processing. What they have found is after they treat the cells with FTI, the cells become, the nuclear shape become normal again, from abnormal to normal. So if you paid attention to the recent news, so the first clinical trial of Progeria, which is using the farnesyltransferase inhibitor, the result is already published about 2 months ago. So what they have found is the farnesyltransferase inhibitors can help. It can reduce the severity of the syndrome and make the patients to be a little happier. But this is way far away from completely cure the syndrome. Statistically, we see improvement. Why? Yeah, it is pretty obvious because the mutant protein is still there. All we are doing is to remove it from one place to the other, according to our working hypothesis. So it will have other functions inside, toxic functions inside the cells. So what we have done recently is we suggested another drug called Rapamycin to be a promising treatment to progeria. Rapamycin is a phase 3 clinically approved drug, and it has been widely used as an immunosuppressant for tissue transplantation. And also it has been suggested to be an anti-aging drug. So what does rapamycin do inside the cell? So it actually gets to the one major cell pathway called the TOR pathway, mTOR pathway. So this pathway censors whether the cells have enough nutrients or whether there is a growth hormone stimulation. If there are, the pathway is activated. Then the cell is started to make mRNA and synthesizing protein. In the presence of rapamycin, or if there is no nutrient, the cells will know, okay, it's time for me to stop making new proteins. The cell will turn on another pathway called autophagy, which will, I view it as a recycle machine. So it will degrade a lot of broken organelles or protein aggregates inside the cells and reuse the amino acids. What we have found with rapamycin is after we treat the cells with rapamycin, first of all, the nuclear morphology looks a lot better, and the cell proliferation is promoted, and the cell life is prolonged for cellular life is at least for 2 more months. We can grow the progeria cells. And again, we use nuclear blabbing as an output of our assays. So we asked, without the treatment, there are 40% of the cells. Without treatment, there are 40% of the cells which is blabbed, and with treatment, only about 10% of the cells they are blabbed. We further went on and showed that with the rapamycin treatment, the autophagy pathway is activated. So as you can see here, with rapamycin treated, you can see the progerin, which is a toxic protein, the degradation is a lot faster. But if you block the rapamycin with the autophagy inhibitor, you block the protein degradation. So the idea is, with the rapamycin treatment, what happens is the rapamycin activated the autophagy pathway, which started to remove the toxic protein from the cell. So that might be from the bottom of the question, to remove the toxic protein from the cells, we can kill the cell and improve the phenotype. with that, I always mention we use the nuclear morphology as a primary output of our assays, both in the FTI treatment and also in the rapamycin treatment. So this is a classical picture we like to show you yearly. So what you can see is in progeria cells, you have 30 to 40% of the blebin, which is abnormal nuclear shape, and in the normal, you see 10%. So probably in your mind, you have something like this, a normal cell and a blebbed cell. Is it true? So I'm actually showing you a daily experiment we are doing. This is a situation we are facing. There will be some obviously abnormal cells, abnormally blebbed cells. I guess you all agree. And then I want to pose a question here. What do you think these 3 cells with question marks? Are they normal or are they abnormal? Are they blebbed? I see people shaking their hands and heads and nodding that yes, they're normal or they're not abnormal. So you can see actually the quantification can be really subjective. And if you think we do a treatment, ask multiple labs to quantify, we could naturally get different numbers. And when I set up my new lab at UMD, so often I was called by my new graduate students, new postdocs, or undergrads, ask me, Dr. Cao, can you come to tell me whether this cell is blebbed? Can we quantify it as blebbed or unblebbed? So what can we do about it? I have a goal. The goal, we want to have an unbiased quantitative assays. And the assay hopefully can be automated and high throughput, so we can do a lot of screening together. In the end, I hope it can be a user-friendly interface so everybody can use it easily. So in collaboration with Dr. Wolfgang Nozert at Physics, so we actually come up with some methods. So in these methods, what we did is we want to outline the nuclear shape with, we first can stain the cells and then we can outline the nuclear shape. And then we can code the nuclear shape according to its curvature. So what you are seeing here is the curvature is color-coded, the positive curvature, which happens most of the time to the normal cells. So the normal cells are mostly cold-colored, like blue or yellow or orange. But what happened to the blebbed cells? They will have a lot of negative curvatures, which we had, it's by the warm color, it's highlighted in red. So with this approach, we started taking a lot of images. For example, here we have, come back, please. So with here, we have actually 100 cells. And each cell is represented by a straight line here. So what you can see, it is a pretty mixed population. Cells on the left are pretty normal, and the cells on the right are not very normal. So it has a lot of red patches, means it is the morphology has some invaginations. And this is a nice way we can actually view 100 cells in a very small space, so for publication. And then if we treat the cells with either rapamycin or a rapamycin analog, we have found, we see the drastic improvement of the morphology. So all these red patches are reduced. And with this quantitative approach, what else we can do? We can do clear quantitative assays, like we can do bar graph, with numbers. Furthermore, I think there was a bonus. After we actually circulate the nuclear shape, I was only interested in curvature, but we got a lot more. We get the area, we get the perimeter, we get the circularity, we get a total about 14 different measures. The communication between all these measures will be important for us for the future understanding of the nuclear shape and aging. So now I have been telling you a lot about the nuclear shape, but our central hypothesis has something to do with chromatin organizations and gene expression. Why I think chromatin organization is important? The reason is actually the first solid evidence came from Dr. Bob Goldman's lab at Northwestern University. What they have done is they did just a straight staining with a histone epigenetic mark. They found in the cells which the nuclear shape still looked normal. What happened to the epigenetics? It's already altered. For example, here you see a lot of foci in the normal cells, but in the progeria cells, it becomes dispersed. And we think these are independent events, and we hope to understand what are the regions in the genome the epigenetics are changed. In order to do that, we use a bunch of assays. So this paper was just published about 2 weeks ago in Genome Research. Now it is online. I will not have the time to going through the details, but I want to highlight a few approaches we used and the model we come up. So we used a method called ChIP-seq. So I'm sure most of you are familiar with it. Using these methods, we will be able to detect the protein and the DNA interactions. We mapped 2 proteins. One is lamin A-C, obviously, because progerin is a lamin A mutant. The other approach, the protein we tested is H3K27 trimethylation, which was the first epigenetic mark Bob Goldman found is altered in progeria. On the right side, what we did is a collaboration with Joe Decker at the University of Massachusetts. So we looked at the chromosome interactions, inter-interactions, and intra-interactions. So the control cells on the left showing you some grid. You don't need to understand what it is, but as you can see, it is a highly organized interaction structure. But on the right side is progeria cells. What you are seeing here is the interaction become a mess. The high frequency interactions are showing you in red, and the low frequency interactions in blue are totally mixed up. That's what happens in progeria. We further did by an EM approach, looked at under the actual resolution, what happened to the DNA, chromosome, and what's their relationship to the nuclear envelope. So what we have found is DNA and nuclear envelope or laminar interactions are present in progeria cells, but they're missing. Are present in normal cells, but they're missing in progeria cells. So all these assays coming together. So what we can draw is we have such a model now. So in the normal scenario, what happens is the chromosomes interacts with a green color. This is a laminar. And these interactions can help the histones to be histone modifying enzyme to come and modifying histone. All this coming together can help to define a well-organized nucleus with open and closed chromatin domains and the chromosome interactions. And in progeria, what happens is at the presence of the toxic progerin, the chromosome and laminar interaction is disrupted. Then the histone modification is absent. They are all correlated. Then in the end, what happens is the open and closed chromosome domains, sometimes people call euchromatic regions and heterochromatic regions, and they are disappearing. So in progeria, all you are looking at is a total mess. So disorganized, collapsed nucleus. This is our current working model for what happens to the nuclear organization in progeria. So I think I've been talking about cells for a long time, so that's only happened. I have to be specific in fibroblast cells, which are the skin cells from the progeria patients. But if you look at the patient, they have multiple different phenotypes in different tissues. We find that most of these phenotypes are coming from the mesodome lineages, so such as skin, bone, fat, smooth muscles. And very interestingly, the central neuron system, the brain, and also the immune systems are protected in progeria. Why it is tissue specific? This is a question we do not know. Another question which has been intriguing me for a long time is why the patients do not develop progeria earlier, like as an embryo, but they started to show the phenotype when they turn 1 or 2 years old. To answer this question, we started in my new lab a new approach called an induced pluripotent stem cell approach. So this approach recently won Nobel Prize of this year by Dr. Yamanaka and Dr. Gurdon. So what happened in details of this approach is you get the primary differentiated cells like skin from a patient. Then you add some transcriptional factors which are specific for stem cells. And these transcriptional factors can reprogram your skin cells back to your stem cell. And then with that, I think the merit of this approach is it definitely overcome the ethics issues of stem cell research. And also it can be patient specific. So if you regenerated the tissue from the same cell, so there is less risk of the tissue rejection. So what we use is after we get the stem cell, we want to drive them to different cell types to understand the tissue-specific phenotypes. In the future, we may want to do drug testing and maybe cell replacement therapies. So this work was performed by one of my graduate students, Haoyue Zhang, and also my postdoctoral fellow, Zhenmei Xiong. So, and they are very colorful. What I'm just trying to show you here from all this is we successfully made iPS cells from progeria patients. That's the bottom line. And all these different colors are different stem cell markers to show they are very much like stem cell. And interestingly, we also find both the wild-type protein lamin A and the mutant protein progerin, they are absent in the stem cells. So that's kind of give us a hint of why it's a late onset disease. So probably in the fertilized egg, there is no lamin A, and during the development, the lamin A, the progerin is getting built up, and to a certain degree, we started to see the phenotype. Then the interesting question come to my lab. I ask everybody there. So we have the stem cell. Which cell type you want to differentiate and learn? Obviously, the first one and the popular one will be the smooth muscle cells. The reason is the patients, they die of cardiovascular disease. And from a very rare autopsy we have from a progeria patient, we see these dots are nuclei. They are present in, this is a vessel section. So they're present on the intimal and the adventitia part, but in the media, there is no smooth muscle cells. And the smooth muscle cells are the major component of the vessels. And if we look at a mouse model in the wild type, you see smooth muscle cells which are standing in red here. And in progeria, they're absent. Why the progeria vessels do not have smooth muscle cells? Are they the cause of cardiovascular death? So my graduate student, Haoyue, started from the stem cell and drive the stem cell to the smooth muscle cells, taking 2 intermediate steps. And as you can see here, they got a pretty pure, very beautiful smooth muscle cells. The question for him is to understand why they die. After many, many months of hard work, so he finally found out the smooth muscle cells are actually, the progeria-induced smooth muscle cells, they are prone to apoptosis. Here are just 2 graphs. One is showing you from 2 different assays. One is from taking a look at mitochondrial potential. You see a significant increase. And we also do the apoptosis assay. You can see in the progeria, you see a drastic increase of apoptosis. So Haoyue is trying to understand now is why progeria has an induced high frequency of apoptosis. And the working model around here is in the progeria patients, apoptosis is turned on, so the smooth muscle cells gets continuously undergo apoptosis, so eventually the vessels is depleted of smooth muscle cells. And the other angle we took is to look at the fat cell biology. we already noticed there is a drop of the body fat in progeria patients. But how and what happens? My postdoctoral fellow, May, she drove those IPS to the fat cell. What she has found is the fat cell has a differentiation problem. So if you look at the upper right panel there, the red signal is the oil. So it's a fat, and it's missing in progeria. And she then later on went on to look at what happened to the fat cell differentiation. So if you know about the fat cell differentiation, it's just a signaling cascade of many, many genes. One will turn on the other and will turn on the other. So May has found that there are some early genes which are not affected. What should I do? And let's don't worry about it. So the early genes is not affected by progeria, but the later genes are. So what you're supposed to see here is a cross. So the progeria actually steps in the pathway, and the early genes are okay, but the later genes are not. So I'm really not a PC person. So back to my last couple of slides on aging. What we learned about progeria and what's the connection to the normal aging? So one question I asked when I was a postdoctoral fellow was, can the Y-type lamin A make progeria without a mutation? So if we look at the Y-type lamin A sequence, align them with the splicing donor sequence, we still find 5 out of 7 nucleotides are the same. So can it be used to make progeria, maybe under certain scenarios? And the answer is yes. So that's one of the qPCR, RT-PCR I did earlier. So what you can see, the white line lines here are the normal samples, and the red lines are the progeria. This is a band of progeria, and we do see a little bit of the progeria in the Y-type samples. It has been sequenced and confirmed that they are progeria. Then later on with progeria-specific antibodies, we can see progeria accumulated in old people, young people, everybody, like you and me, but at a very low level. So what is the control of progeria splicing in normal aging? That's what we have found recently. So we found that when we are getting older, our telomeres are getting shorter. And when the telomeres gets impaired, the progeria splicing from the normal sequence is activated. So that's the model we have here, is when we are getting older, the telomeres are shortened, either shortened, uncapped, or damaged. Then they will activate the traditional pathway as well as progeria and other alternatively spliced genes. Then all these together can contribute to aging. So in summary, what I have told you today is I have told you our central hypothesis on the nuclear structure and the chromosome organization and gene expression. And I told you we use a stem cell approach to study the tissue-specific defects of progeria patients. And I also told you what is the potential connection of progeria, this rare syndrome, and aging, and what's the telomere probably has a role up there to activate progeria splicing in normal people. So with that, I want to thank the people who did the work, actually. I did not get the chance to talk about a collaboration I had with Dr. Hanahani. These two fellows, Radho and Kun, we are working on the lamin A regulation in aging and RNA splicing in aging. And I did talk about the work Jason did with Dr. Wolfgang Nozert on the nuclear morphology analysis. And Dee is the person we collaborate with Dr. Zhongqi Liu on the plant aging project. And she also has a very beautiful poster this morning on the study of the lamin A processing. It's a cell biology study. And Hao Yu and Mei are the people doing the stem cell research work. And Melissa, I didn't talk about her work, and she is doing a beautiful screen of another clinical approach for progeria in collaboration with Dr. Francis Collins. So we are screening through a lot of molecules to see who will be a next round of the potential treatment for aging. And I also want to thank my funding agencies. Thank you very much for your attention.